So good morning, everyone. I think we will go ahead and get started. It's Monday morning. We're supposed to start at 8:45, so it's 10 to 9. I hope more people that have signed up will start, will continue to trickle in, but we do have an agenda to go through, and we should begin. I would like to welcome everyone. I'm Lida Anestidou. I have the privilege of being the staff officer here at the Academies associated with the Roundtable on Science and Welfare in Laboratory Animal Use, which is actually the parent committee within the academies that oversaw the development of this workshop. It is actually the fourth one we do in our 2 years of operation, and I think it has become to be the most ambitious one simply because of the developments that occur in rapid succession. So, genome editing has been happening for decades. Scientists have been doing this in live cells, in animals, in plants. But I think what's new is the rate of efficiency that is exhibited by some of the genome editing proteins and the increased ability for precision and the potential low costs associated with that. And that may be the fact that allows these technologies to be so much diffused in the research community globally, not just in the US. So can I have the second slide, please? Or do I do it from here? Oh, thank you. I wanted to just put these up on the wall for you to simply show that our workshop, which is the first one, and it's happening today and tomorrow, is one of a number of activities that are happening within the academies. There is an ongoing study that is looking into gene drive research that is expected to complete around April, May 2016. There is another study that is looking at genetically engineered crops, also expected to complete and issue its recommendations again in 2016. There is another study that began just last week looking into social, medical, ethical considerations about human gene editing, following from the international summit that was here at the academies also last week on human gene editing issues. The next slide, please. So as the committee was working to put these the workshop together, and as I said before, the fastness, I mean, it's the quick results that one gets by using CRISPR-Cas9 particularly made it very clear to us that there was a need to discuss not only the science but also the ethics behind all these new organisms that can be created, that are being created. Since the planning of the workshop began 8 months ago, we've had new dogs that were created that had more muscle mass. We've had pet micro pigs, so pigs that are really, really very small and are being sold as pets. And just last week, there was a paper, PNAS, talking about mosquitoes that were altered using gene drives to not carry Plasmodium falciparum. So the research is outpacing everyone. And so while we are looking at the ethics and the science, and we will have both a summary of the discussions issued, and we will put everything online, the presentations, the videos from the webcast, I wanted to also bring to your attention that at this time, tomorrow things may be changing. I'm sure more people, more organizations will do things. The Nuffield Council on Bioethics at the UK has a whole program that's looking on genome editing more globally. And the last issue of EMBO Reports in November had a number of articles that were dedicated to different facets of gene editing. Next slide, please. And lastly, for those of you who are on the web, this is the email that you can send your questions to, and we are very good about having roundtable members, committee members to monitor what's coming in and having the speakers address. We have a Twitter handle, please tweet. It's very nice to see these kinds of things in the Twitter world. And I also wanted to remind everyone that this is an open meeting. and everyone is welcomed. And this is webcasted, so by virtue of being here, there's a consent by all of us that that is fine. And as you know, all the meetings at the academies are free and open to the public, and they are meant to be a place for congenial discussion, even disagreement, but always congenial and always polite. So thank you very much for coming. And it's my pleasure to introduce the co-chair of the committee that put this workshop together, Dr. Rhonda Wiler.
Thank you, Lida. And welcome everyone, both here and virtually, to this workshop. And I'd also like to thank the other members of the Roundtable for their support and contributions to this meeting. And a great thanks go out to Lida and her team for their extraordinary ability to pull this all together in a relatively short period of time. So the focus of this workshop will be primarily on how CRISPR-Cas9 is transforming the ability to modify the genome of any species and bring awareness to how CRISPR-Cas9 is being implemented in a variety of animal use settings. The gathering is only a beginning of a longer conversation covering a myriad of questions, such as what are the efficiencies and obstacles to molecular editing of animal genomes using this new technology? What is the potential effects of CRISPR-Cas9 will have on expanding the number of animals and variety of different species that can now be entered into biomedical research? What parameters need to be in place to safeguard the humane and ethical care and use of animals, both in the editing phase of their genome as well as the productive and procreation phase of their life? Should this technology be used across all animal species? What ethical barriers should be considered before moving forward? And what would our obligations be towards the quality of life of these animals? This session presented over the next 2 days are intended to begin these discussions. Once again, a novel technology that has the potential to impact future generation is pushing the debate on the moral and ethical considerations of the ability of humans to modify the evolution of life back into the forefront. In this case, CRISPR-Cas9 provides unprecedented power to precisely alter the genome utilizing reagents that are relatively simple to design and synthesize. Combining the power of CRISPR-Cas9 with the technological advances in sequencing and reproductive science is a trifecta that has caught the attention of many. This is a new frontier, and I hope the content of this meeting will engage many more in this conversation. So with that, I'd like to introduce our keynote speaker this morning, Dr. Peter Hohenstein from the Rosalind Institute. And his talk will be on the age of editing, trends and techniques in animal transgenesis. And this will kick off this important discussion. With that, it's a great pleasure. I welcome Peter.